started, and Adam is going to be providing an introduction today.
Yeah, so good afternoon, everybody. We are pleased to welcome Dr. Melissa Haendel, both to OSU and to our college's seminar this afternoon. Melissa is the newly appointed Director of Translational Data Science in the CGRB, the Center for Genome Research and Biocomputing, here at OSU. And prior to that, she was up at OHSU as the Director of the Center for Data to Health and as an Associate Professor in the Department of Medical Informatics and Clinical Epidemiology. Her Ph.D. was earned from the University of Wisconsin in neuroscience, and her research interests include precision of rare disease diagnosis and using informatics to reduce healthcare inequities. And today she's going to present an open science approach for rare disease patients.
Thank you so much.
Testing? Okay, I think I can. So thanks very much. It's really exciting for me to be here because OSU is actually my alma mater from my postdoc so many years ago. And so it's really exciting for me to come back and be faculty here now at OSU. So back in the day, I was much more of a molecular toxicologist and not so interested in public health, but my meanderings since then have brought many different areas of research together. And I wanted to put together a talk today that was really going to kind of highlight some of those things to kind of bring a perspective to public health that research that maybe is maybe a little less common or less familiar. And that's really an integrative one where we really bring different types of data and different kinds of research together in order to inform rare disease diagnostics in particular. So one of the problems with diagnosing rare diseases is that, well, first, they're rare, so we tend to not have as much knowledge about sort of the molecular mechanisms, the specific for Mendelian diseases, the specific molecular lesions in the genome that are responsible for causing the disease are really difficult to identify. And so there's a variety of different diagnostic pipelines that essentially perform whole exome or whole genome sequencing to help support the identification of those variants that might be responsible for the patient's rare disease. But unfortunately, a lot of times those pipelines tend to result in hundreds or thousands of potential variants that we don't know anything about in the human. So if we think about our patient over there, we're really we're also the whole genome or whole exome information to try to figure out what is the molecular cause of their disease. We tend to use increasingly more, but less about their actual phenotypic information. We certainly don't use very often kind of large-scale metabolomics or other omics types of phenotype information. And then as we get down into socioeconomic factors or environmental exposure issues that may actually have effects on rare diseases, whether or not they're Mendelian or otherwise. And so really what we're trying to do is kind of compare this picture of a patient, which of course is also temporal in its nature, especially where socioeconomic factors or environmental exposures are concerned, against some reference population over here where we have a genomic reference or we have disease definitions, we have population frequencies for the variants, we have population-based statistics and disease risk factors and some information about exposure information, and there's some wonderful research here at OSU that helps to try to correlate some of these things. And so really the challenge is that we just don't have the power to diagnose very, very many of the patients because we don't know how to compare those things over there about the patient against the reference information or how to incorporate them together with the genomic information. So one of the things that we had conjectured many years ago is that inclusion of information from model organism species would actually help us diagnose patients. So if you look at the number of human coding genes in the EXEC database, which is essentially a large-scale variant collection of population frequencies for variants, roughly half of them have phenotypic coverage. So around 9,739 of those genes have something that we know about the phenotype. So if you're a rare disease patient with a mutation in one of these genes, we can't help you, right? So when we take the orthologs of those genes, so those 19,000 genes that are coding genes in the human genome, and take the orthologs of those in the 4 most commonly used species, zebrafish, the worm C. elegans, fruit fly, and mouse. So we have those same 19,000 genes in those 4 species. Collectively, they cover 14,000 of those genes have phenotypic information associated with them. So now when we take the union of those, we end up with, we go from around 51% coverage of the human coding genome to 89% coverage of the human coding genome. So now all of a sudden that we've increased our power to actually potentially reveal what the molecular cause patients' phenotypes might be by comparing phenotypes against model organism species. And as many of you may know, model organisms that are used in research to model disease, but what you might not know is some of the plethora of other organisms that are sort of non-model organisms, things like, you know, the armadillos being the natural host for leprosy mycobacterium, which is the only other species besides humans. So we necessarily have to be leveraging all the species data in order to help us understand the molecular causes of disease in humans. We did actually a little survey of the literature and mined more than 4,000 abstracts from PubMed, and actually just are doing a scan for all the different species that are used. And this is very preliminary work, but what we found is that you can really look across all the different taxa to determine which species are being used to model which kinds of phenotypes in human. And it's actually incredibly diverse and growing for all species, not just traditional NIH style model organisms. One of the challenges, though, is that we have to be able to tell the computer what things like palmoplantar hyperkeratosis actually means, which is a clinical phenotype term, but the computer doesn't really know what to do with that. How do we use that in the same way that we use an A, T, C, and G when we're comparing sequences in molecular data? And the problem is even more challenging when we think about the different communities. So that same phenotype might be similar to ulcerated paws in the mouse, and a patient might just say they have thick hand skin. So these are very different words to describe similar phenotypic features. As it turns out, each community also tends to develop their own vocabularies for representing these things. So in human, we have the human phenotype ontology, which I'm going to talk to you a lot about in a minute. But in mouse, we have the mouse phenotype ontology. And it actually, this is just a sprinkling of the hundreds and hundreds of terminologies that have been developed by the different model organism communities. We have zebrafish terminologies, we have yeast terminologies, we have mouse terminologies, we have terminologies for the pig. The pig one's especially interesting. There's things like bacon fat depth as descriptors for their phenotypes. So what we've done is we've actually created a logic-based system to describe these phenotypes in a way that the computer can understand it. So we take that term, palmoplantar hyperkeratosis, and we break it down into, what's really, logic statement that the computer can run software tools called reasoners over to understand something about what can it know about the term palmoplantar hyperkeratosis other than the label that it has. And so what that looks like in sort of pictorial form here is we state a logical statement that palmoplantar hyperkeratosis is equivalent to an increase that's located in the stratum corneum layer of the skin that's part of the autopod. And these terms here all come from a species-neutral set of vocabularies, such as the gene ontology for terms like keratinization, or the Uberon anatomy ontology for these two terms. And this logical statement can be related in an exact way or in an inexact way using logic to terms like ulcerated paws. And so when a mouse researcher used this term, we now know that this term is actually similar to this term, and we can actually create that equivalence across species. And then if we have information about the molecular nature of the lesion that's causing ulcerated paws, we might guess that that might be the gene that's responsible for causing that human disease. So back in 2009, we published the first paper showing that this approach could work. Essentially, for those of you that are familiar with sequence analysis, a phenotype blast. but based where we have no knowledge of the actual underlying sequence information, we could retrieve, based on human phenotypes such as the cornea being opaque and the iris being absent, we could compare against all known diseases. We would compare things like cornea being opaque to, these eye phenotypes, eye decreased size, lens being fused to the cornea. Over here for the zebrafish, for the retina being malformed, and over here in the fly, eye absent. We were able to return all of the orthologs of the gene that we knew caused those mutations, caused those phenotypes in the human based on the phenotypes alone. At the same time, one of my now very favoritist colleagues was developing something called the human phenotype ontology, which was a new taxonomy for phenotypic abnormalities, things like deeply set eyes being types of abnormality of the globe location, which would be types of abnormal eye morphology. But at the time, this was a clinical vocabulary being written by a clinician and didn't have any of that underlying logic that I had just shown you as that sort of compositional logic-based approach. So we combined forces, and we've now added all that logic to the ontology so that you can say things like, hyposmia, which is a lack of sense of smell, is a lack of sensory perception of smell. And this term from the gene ontology has been annotated to 34,000 annotations in 22 species. So we know a whole lot about what it means and the molecular nature of a lack of sensory perception of smell across, all of life. We can now use that information to inform patients that have a lack of sensory perception of smell. People often ask us, why do we need another clinical terminology? The other thing that's different about this phenotype ontology, this HPO, is that the human, like we describe a mouse or any other biological subject in any other observational setting, which is very different than a clinical vocabulary, which tend to be used for billing purposes or quality of care evaluation and those sorts of things. They're not designed to describe the patient sitting in front of them as a biological subject, so they're therefore not as good at revealing the underlying biological mechanisms of disease. And when we compare the human phenotype ontology against all the other clinical terminologies, some of which are shown here, but all of which are included in the United Medical Language System, it's more than 50% of the terms don't occur in any other clinical vocabulary. So the things that we're describing about these patients, things like lack of sensory perception of smell, are just not in the clinical vocabularies. So what do we do with this? What we do is we describe the patient in terms of a set of terms that describe the phenotypic profile of the patient. So here's your patient up here. They might have a term like hypotonia or having a flat back of the head. We would want to then try to match these terms against a known disease definition where we've curated a kind of a gold standard set of disease definitions that are the set of phenotypes that are known to occur with a given disease. But one of the challenges is that non-exact matching. So what if our standard definition has an abnormal skull morphology or a decreased muscle mass? These are not going to be exact matches. And it's not a string matching problem, it's a conceptual alignment problem. You as a human being might know that hypotonia would be related to decreased muscle mass, but the computer doesn't know that. So that's one of the challenges that we've aimed to overcome. So I'm going to show you a silly animation, but it's seemingly the only way that I can actually get across what the algorithm does to try to match this. So if you think of the patient then as a set of nodes in a graph where each node represents one of those phenotypes that I described earlier, so hypotonia, abnormal sensory perception of smell, where the dark ones are the ones that are representative of that patient's set of phenotypes or phenotype profile. We can then match them against what we know about model organisms or non-model organisms, as well as our gold standard disease definitions. And so what happens then is if we look for the most similar match, here we try to do a graph matching algorithm that will try to align the patient's graph with every other graph that's a representation of the phenotypes of that organism, be they a mouse, a zebrafish, or another human disease definition. And you can see how the non-exact matching works. So over here, where we might have had a term from the patient here, over here we might have had a term from the model. This is the common node in common. We have a number of different graph metrics that allow us to kind of tweak this and understand what's the best closest match in terms of all the phenotypes as a collection. One of the other challenges that we have, so that's lovely, but these things all hang off of a disease definition. What is a disease? Well, the reality is that we don't actually define disease very carefully in informatics. This is an example of 4 different resources being combined together with they all sort of cross-reference each other. So in a clinical terminology would cross-reference another clinical terminology to just sort of say this disease is related to this disease, right? But there's not really a good representation for what that means. And so when you combine them all together, which we have to do because we're combining data from very, very many sources in order to answer this question for how to diagnose the patient, you end up with this spaghetti craziness. Where there's a lot of different relationships that can kind of piecemeal together a big picture of a disease classification. But you end up with terrible inconsistencies. So, for example, here when we look at all the sort of terms from all the different vocabularies related to hemolytic uremic syndrome as a disease definition, remember, these are the terms that our sets of phenotypes are related to. So eventually a patient's going to get a definition, a diagnosis of this disease based upon our algorithmic approach. It's not going to work if these diseases are all coming from all the different data sources are all inconsistent. And here what's happened is that when we combine these different sources, we end up with atypical hemolytic uremic syndrome being equivalent to typical hemolytic uremic syndrome. And so what diagnosis should we give the patient then, right? So it's a huge mess. And it's a mess because fundamentally the way that we've described diseases over thousands of years really is, based on different types of perspectives. What kinds of treatments work? What's the etiology? What's the molecular mechanism? And the more we know about molecular mechanisms, the more how we define diseases is changing. Similarly for environmental factors and diet and exposure and socioeconomic factors. That same first slide that I showed you. So this is a very quickly changing landscape, and that's really what precision medicine is all about, is changing the way we define these things. But in the meantime, we're stuck with these limited informatics resources. And so there's actually a group called the ClinGen group that we've been working with to, how many of you are bird watchers? Not too many. Well, if you're a bird watcher, you're familiar with the lumping and splitting problem, which is where you get really excited when species are lumped because it means that you can say you've seen that species if you've seen one other subspecies. Or maybe if you're a splitter, you're excited when they're split because you can go off to some other part of the country and go see the other half of that species that is now a problem with these diseases is how do you define when a disease is a new disease versus when it's the same disease as another disease? And so this group is working on this kind of a classifier for deciding when do we create a new disease entity. And so we have this unified approach to managing all of this, and that cleans up that big semantic mess. So now we have a tidy representation of diseases that we can hang our phenotypic profiles that are in those graphs for the patient. Once we've done that, we can now combine that data with the whole exome or the whole genome information. So this is how this works, and it's a tool called Exomiser. And Exomiser takes all the public data that we have for reference whole genomes, variant frequencies, combines it with pedigree, inheritance model matching, protein predictions, and these are all fairly standard kind of whole genome sequencing efforts. But the special sauce is really then combining that with the phenotype data. So over here, we take data from hundreds of resources, such as Orphanet or the online Mendelian inheritance in man and human, but also the model organism databases, such as the mouse, the zebrafish, fruit fly. And then we also use a lot of protein interaction networks to crosswalk so that we can do guilt by association, so that when something looks like it might be the right thing, but it's not coming up via the methods over there, we might prioritize something it's talking to in the protein interaction network. And so then using all the protein interaction data and the phenotype data, we can combine it with the whole exome data and come out with a prioritization for a patient's molecular lesion for their disease, and hopefully come up with a diagnosis which is necessary in order to identify potential treatment options, among other things. So this is what this looks like. This is an actual case. These are two patients a 3-year-old girl and a 14-year-old boy who actually came into the same clinic roughly 2 weeks apart, and the clinicians, world-class clinicians in rare disease diagnostics, did not recognize that these two patients actually had the same disease. And this really gets after this idea of lumping and splitting problem as well. So in this case, they were both diagnosed using this Exomiser tool and this phenotype matching-based approach with Wiedemann-Steiner syndrome. But what you can see is that not all phenotypes for this patient, for the boy, are present in the little girl, but they all have some match to the gold standard set that we have here in the middle. So for the gold standard set, we have short toe, which matches long toe over here in the patient. And you might say, well, that's not a match, but that's exactly what we're talking about. In this case, this patient had a toe phenotype that's not exactly the same as the gold standard. Other cases are, of non-exact matches are short middle phalanx of the finger, the cone-shaped epiphysis of the phalanges of the hand. Well, those are both related to, finger phenotypes, but are not exact matches. And you can see there's other opposite phenotypes as well. Macrocephaly versus microcephaly both match the gold standard term microcephaly. In one case, it's an exact match. In the other case, it's an opposite, right? And so, but this was the best match for both of these patients in terms of that phenotype profile similarity in the graph. And they were both diagnosed with Wiedemann-Steiner syndrome. You might ask, well, maybe that's not the same disease if they have these opposite phenotypes, so it's lumping and splitting problem. Turns out they have two different variants in the same gene, one of which causes slightly different phenotypic presentation than the other one. But currently, the disease definitions suggest that those two variants are still classified as the same disease. So it's a lumped disease in that case of Wiedemann-Steiner syndrome. So here's an example where we were able to help diagnose a family with a mutation in STIM1, which is a platelet disorder based upon phenotypic similarity to a mouse, where we had no information at all about this gene at the time in humans, so nothing in any of the clinical databases. So that was really exciting. It was the first case of being able to use the mouse data for a diagnosis. We're actually running this tool right now in collaboration with Genomics England. We've gone through 19,000 families that have been diagnosed, and 82% of the variants that have been identified in these families now were in the top 5 hits across a wide range of families. So we've gone from thousands of variants to the top 5 using this tool by combining data from all these different resources. The other thing that this approach has really helped us do is to reveal new disease genes. So this is the International Mouse Phenotyping Consortium, where just by doing this phenotype comparison against Mendelian diseases for which we don't know the molecular basis, comparing them against this mouse knockout program where we're knocking out every gene in the mouse, and comparing the phenotype data that's structured in the exact same way as I showed earlier, we're able to reveal new candidates for what are the molecular basis for these genes that for these diseases that we knew were Mendelian for a long time but didn't know what the molecular lesion was. And so this is an example with Diamond-Blackfan anemia, where the phenotypes were similar based upon increased mean corpuscular hemoglobin and decreased erythrocyte cell numbers and differential expression of these genes. And so this, based upon some early evaluations, may account for about half the people that have this disease. We've now identified the gene for that particular disease based on the mouse phenotype. Another really cool thing about this is many families with rare diseases have multiple rare diseases running in one family. And this was a fantastic paper that came out early 2000, showing the use of this approach to tease apart, based upon the phenotypes, multiple different molecular lesions running in the same family, so that if you have the full pedigree, you have a profile for everybody in the family, you can actually tease apart for a given proband for the patient that's presenting, you could tease apart that they actually have two diseases based upon similarity to their parental lineages and the phenotype profiles that those parental lineages have. So that was very exciting, and there was quite approximately 4.9% of the exomes that they analyzed had two diseases or more. So I want to switch gears a little bit and talk about a patient engagement project that we've been working on, and I think this is really where it kind of hits the public health realm and moves from just an informatics tool that your clinician uses that you don't know anything about to actually like being part of a patient's life. So we want patients to be able to tell us about their phenotypes as well. When a patient goes into the clinic, the clinician does a bunch of different measurements and analyses and clinical evaluations and laboratory assays. But they don't often ask, is your baby inconsolable, or do you snore at night, right? These are phenotypes that happen in your daily life that are not clinically observed. And unless you as a patient happen to mention those things, your clinician may not know between rare disease patients who often have very long odysseys to be able to capture this information and understand what is the clinician going to do with it in more of a collaborative fashion. So to support that notion, we actually went through the entire 14,000 term set for the human phenotype ontology and translated roughly a third of it into what we called laypersonese, which were really patient-accessible terms that they could use the same terminology as the clinicians are using to describe their phenotypic characteristics. So some example here, might be hypertelorism, which the patient would see a term for wide-set eyes or widely spaced eyes, or cyclopia, which would be a single central eye. So these are the eye region. And so we were able to get about a third of it where it wasn't just clinical terminology that we couldn't translate. Here's some more examples and how they compare. So, for example, impaired pulmonary function might be trouble breathing, or arachnodactyly might be spider fingers, long fingers. And we scraped a whole bunch of different websites and patient groups and things like that to find as many terms as we could, and we're constantly improving that. So roughly 4,500 terms have these synonyms. There's roughly 7,600 terms. So it's roughly 35% of the coverage, and that covers about 60% of the diseases of all diseases that we've annotated these profiles to. And so the question then we have, and this is an ongoing study, is how diagnostically useful is a layperson version of this ontology. We want to know how close can a patient get to, to helping with the diagnosis. And so we also have a survey that's currently the standard of care for patients that they fill out in a rare disease setting that was developed by the Genome Connect group. And this survey, just is a body system survey that says, do you have any troubles breathing? Do you have any, facial features that you find that you think are unusual, and that sort of thing. And then we have this layperson version, and then we have the clinical version. And we would hypothesize that the clinical version would be the most diagnostically capable than the layperson version in the survey mapping, because there's a mapping to just only a few hundred terms in the ontology from the survey. And so what we've done now is to simulate a self-reported profile for 7,700 different diseases. So for every disease, we created a kind of synthetic profile that's a subset of the clinical one that's just the layperson version, or the Genome Connect survey version, which only has a few hundred terms. So you can imagine that's pretty small. And then compared that against the reference gold standards that we use currently for diagnostics. And so roughly, so it's 4,500 terms with the layperson synonym, 216 from the survey, and then looking at the semantic similarity to our known rare diseases. In this particular analysis, we had 7,600, and looked at what's the drop in sort of how good the annotation is and what diseases are enriched. And so here's kind of what that looks like for a real example. So over here, we have a nephroblastoma gold standard set of annotations, and you can see that the layperson version here, we might have polyuria, it was an increased urine output. Neoplasm of the lung is a lung tumor. Some of them are an inexact match, so where we didn't have a layperson match for a given term, we would just go up in the tree to something that did have a layperson term. So in this case, the patient would have recorded kidney cancer rather than nephroblastoma, for example. And you can see that some other examples here. So lymphadenopathy is related to abnormality of the lymph nodes, which is a higher level kind of term. But many of them are actually exact matches to what the clinician would actually use. For the survey, the mapping is quite high level, so they almost all are inexact matches. So, for nephroblastoma, it would be a renal neoplasm, or for neoplasm of the liver, they would just be abnormality of the liver. For hematuria, it would just be a urine homeostasis kind of problem. So here's the results. Again, these are the number of terms that we have in terms of how much we can use to describe the patient's diseases. And what the results show over here is really actually very interesting, is that 80 to 90, roughly half of the diseases were showing up as top ranked using just the layperson version. So that means that for roughly half of the diseases, if a patient filled out their layperson HPO profile perfectly, that roughly half of those patients, roughly half of those diseases would show up in the top 10%. So that's incredibly good coverage, and we were expecting it to be much lower. What's also interesting is even those 200 terms, got us higher than we expected. Roughly 50% of the diseases turned up. Now, one thing to note is that just because they turn up in the top 10% doesn't mean that they're not a whole bunch of them ranked the same. So what we're losing is the differential diagnostic. So they're getting them to the top because they are really similar, but there are also a lot of other ones that are really similar too, and that's where the clinical collaboration would come in. The other interesting thing is that when we look at the number of diseases by performance category, we looked at whether or not the human phenotype ontology outperforms the Genome Connect survey, whether or not they're equivalent, and whether or not the Genome Connect outperforms the human phenotype ontology. And we actually had a few cases where the Genome Connect survey was outperforming the human phenotype ontology. And so why the gaps? There's a couple reasons for that. One is that some of the terms that should have a layperson should be layperson eligible, so to speak, like schizophrenia, but were not designated as such, so we were missing some that should have been there. Some which weren't really layperson at all, such as abnormal EEG, which is maybe not really a layperson term, or some that really don't distill into short lay-friendly labels. Some of the terms they're just really long descriptions and were untranslatable. So we also found that some diseases were enriched for being better capable for the patient to have a good relevance ranking. So things that had developmental defects during embryogenesis, dysmorphologies, things that had bone diseases or connective tissue diseases, skeletal things. You can imagine things that are clearly observable by a patient outwardly. Things that didn't score so well, I don't have a slide for that, are things that required a lot of clinical labs. So revealing, glycan homeostasis is not something that a patient necessarily would know unless they read their labs, which many, of course, they do. So we have a new tool that we've been developing for patients to be able to self-phenotype themselves, and we're going to start a human study on this tool and compare it to the standard of summer. And our hope is that this tool will really, promote patients to be able to collaborate with their clinicians in helping them diagnose their rare diseases. So just to kind of summarize this part. The patients, we believe, are really a key source of phenotypic information, and we want to kind of promote this collaboration. For over 95% of the 7,000 plus diseases, the layperson phenotype subsets were more diagnostically useful than the survey-based approach. So we're really excited about that because at least we're going to advance beyond that standard of care. And then some diseases had this low coverage and require, more of a clinical lab-based diagnosis from a clinician. So an example here would be glutathione synthetase deficiency. So now we're going to test how confident we are in the diagnosis. So trying to get some measure of how many are ranking at the top at the same level and how differentially diagnostic it can be. And then also what's the confidence, and then adding noise and perturbation, because the patients are not going to fill out their forms, their profiles exactly correct. So we want to know what that's going to look like ahead of time before we test it in the patients. Okay, so I'm going to move on to the next topic, which is how do we actually use some of this information more broadly? And so this is an effort to create a new standard for phenotype data exchange. So we have lots of standards for genotypic information and exchange, such as, BCF files and bed files for whole exome information. We don't really have good standards for moving phenotypic information around. And so this new effort was started. It was called PhenoPacket, sort of jokingly, because it was I just want to have a packet of phenotype information that I could pass to a journal or pass to a clinic or have a patient generated. And so it stuck, and that's what it's called now. So the idea of phenopackets is to have this kind of just exchange format where we can represent these phenotype profiles. It could also include, quantitative molecular information, and so be able to associate that with genomic information and pedigree information for Mendelian diseases, but also increasingly including other things such as environmental exposure information. And so the idea is that, patients, families, physicians, basic science researchers, could create these phenopackets, and these could be used by patient registries and clinical trials for submission to journals. We can then scrape them out, and using a variety of tools, we can then kind of integrate these, which would really improve the idea of cohort identification for creating trials for rare disease patients. It's really hard to do clinical trials in rare disease patients because they're so hard to find across the globe. And also patient matchmaking, finding the N of 2 patient means everything to a rare disease. Once you find the N of 2, some clinicians argue that that's the requirement for actually having a diagnosis, is that there's more than one family that has the same molecular lesion. With the long-term goal of course, speeding diagnosis up and also revealing mechanisms so that we can come up with treatment strategies. So here's what a phenopacket actually looks like. So here, this is a family with an NGLE1 mutation. Here it's just encoded as patient 16, so it's de-identified. And you can kind of see that it just kind of shows these phenotypes. Allorecmia is an inability to produce tears at birth, and it's congenital onset. And here we've included a source which is from a PubMed ID. And so these sorts of information can then be used by clinical labs and public databases and journals to have a computable representation of that phenotype information, so that when the next patient comes along that has that very unique set of phenotypes, we'll be able to match them. We also want to be able to enable patients to be able to excel. So right now, what many rare disease patients do online is they go to Facebook and they type the phenotypes, and they try to find other patients that have the same set of phenotypes, or they go to Patients Like Me, which really doesn't support rare diseases very well. And we actually work a little bit with the Inspire organization, which is trying to do a much better job to help create communities for rare disease patients. But patients should be able to create this in a more structured way as well. So instead of saying allorecmia, they could say dry eyes, and the system would know that those things mean the same thing when we try to scrape the web and find other patients. And so these could be used for patient registries and social media. And then I finally want to end on just some work that's going on to actually create a global framework for finding the N of 2 patients that doesn't rely on Facebook, which is probably a good thing right now. So basically, this is called the Matchmaker Exchange, and it's part of the Global Alliance for Genomics and Health, which is a global grassroots community that aims to try to support all kinds of different genomic health in all different fields, but also including rare disease. And for rare disease, we have a clinician and a patient. They have a phenotype profile. They have a set of candidate variants that we don't know which one it might be. They may be an N of 1 patient. And so we want to query other clinical sites to try to see if they have a patient like mine. If we have a patient like mine, as I suggested earlier, that means everything, because it means we can confirm that that molecular lesion is responsible for the phenotypes that are being seen in the patient, and that opens up possibilities for treatment. We can also query all the model organism databases, and in our program we provide services to all the clinics to help query to make sure there's not a matching model organism that would also help. But then finally, there's the ability for patients to be able to contribute to this ecosystem as well. And I don't have it on the slide, but there's one of the services actually allows patients to create their profiles and post them up and try to match themselves against other patients globally. And what I'd like to do is, this is a very closed system, but it's because of security for identifiable patient health information. But the patients are already posting this information like this on the web anyway. When you're a rare disease patient, you don't really care so much about posting information at this level of detail. It's very high level. It's not, it's not your social security number or your, nuances of your particulars. It's really just the biological aspects of your disease. And so we're trying to create systems now that would allow patients to just post these sorts of things much more openly and not have to rely so much on a network like this where not everybody in the world's on the network. We want to use the World Wide Web instead of the Matchmaker Exchange in order to achieve that matchmaking. Okay, so just to conclude, so many diseases present with very distinct phenotypes, making computational description of those phenotypes a useful tool for diagnosis and also for revealing mechanisms and identifying treatments. The human phenotype ontology can capture the structured information about those phenotypes and currently contains actually about 14,000 classes now. We just added a whole bunch. For phenotypic features that are really biological attributes of the patient's symptoms. And then we believe that patients are a key source of this phenotypic information and should be participating in a collaborative fashion to maximize the diagnostic capability that those families might get a diagnosis. And that we should share the phenotype information alongside genetic data to support the diagnosis and discovery, to create patient communities, and also for reproducibility in the literature, to be able to better mine the literature and require less burden on those of us who try to get information out of the literature in order to create such diagnostic tools. And so I wanted to thank the many data resources that we use. I don't know if any of you are familiar with the concept of research data parasites, but I'm definitely one of them. If we could not do any of the work that we do without being able to use public open data sources in order to create these diagnostic tools. So there's many open data sources that are used. And then also, of course, all very many people that I work with, but in particular, I'd like to thank Peter Robinson at the Jackson Laboratory and Christopher Mungle at the Lawrence Berkeley National Lab, who've been my partners in this work for over 10 years. So thanks very much.
Questions or comments?
Yeah.
So how do you connect that are filling out these phenotype surveys? Is that like a PCORI project?
It's a PCORI project. Okay. Yep. And we have
research networks?
Yep.
Okay.
We're not using the PCORI data models because those models don't really represent rare disease, and we're not really using the clinical data in that way because it's patient-provided information. But it is funded by PCORI, and we have patient advisory board helping us design the tools and giving us the feedback that we need to make sure that they're going to work. And then we have kind of a series of, we have a couple different rare disease cohorts that are all, they all have a diagnosis, and we're confirming the diagnoses in the EHRs before contacting them, but then they will be filling out their own phenotype profiles. And some of those studies also involve some qualitative interviews to help understand how they feel about filling it out so we can better tailor it to make sure that it's, feels engaging and doesn't feel, threatening or anything like that. So, yeah.
Very exciting. One of the things that strikes me about it is it's the standards upon which you're comparing are probably also changing too.
Mm-hmm.
How do you deal with the sort of, dynamic environments of what you choose as a gold standard?
That's a really good point. We do, because we're research data parasites, can't do all the curation work of the literature ourselves or have all of the sources, but we do a lot to try to clean things up. So like that slide with the messy disease definitions, everything is really strictly versioned. So we version all the sources, we version all the things we put together, both the data and the semantic structures, the algorithm. And so if everything is versioned, we can at least keep track. And so we'd know, like, if, and actually as we move more in, right now we're sort of a research grade tool and we're trying to move it more into a CLIA certified clinical grade tool. If we ever get sued for giving somebody the wrong diagnosis, we want to be able to point back and say, well, the knowledge at the time then was this, and here's all the pointers to that information. And we also want to be able to for many patients that don't get a diagnosis using this suite of tools and other tools, you want to rerun it on a regular cycle so that when new knowledge comes in through one of those sources, be it a clinical source or a model organism source, that there would be an update and the clinician would be able to tell the patient, hey, there's some new information, let's evaluate this new variant that comes in. And that's where the strict versioning really is important as well. So, yeah. But it's challenged because it's dependent on us collectively as a community to curate this knowledge and get it out there as quickly as we can. And we're trying to get, so one of the things is for all of the new N of 1 patients that get diagnoses or, small cohorts, creating new records for them so they can be used in the diagnostic tool Exomiser, so not waiting for the publication. So essentially bypassing publications for those rare patients so that we can bootstrap diagnosis globally. And so we've been working with Orphanet in order to create those files that can be used before there's any public knowledge about those findings. Yeah.
I'm wondering, so obviously I think this is really fascinating stuff, and I wonder how well the models work for things that are more environmentally based rather than genetically based.
That is a very good question, and it's one of the reasons I came back to OSU. We're just starting to work on some of the similar types of approaches for representing environmental information that might be important for some diseases. So for example, for Ehlers-Danlos syndrome, we have a pretty good indication that there's a lot of diet and also life stage, so pregnancy or menopause are very have these effects on the phenotypic variability of Ehlers-Danlos syndrome. How do we represent those things so that when somebody comes in that hasn't been yet diagnosed with Ehlers-Danlos syndrome, we can take those things into account as well. And so we're really just starting to figure out ways of representing those. But I think it would take a similar approach to the sort of modular representation. One of the challenges there is really how do you combine those data, that representation? So we use these representations to bring data together, but the data that we need for environmental data is very diverse and large. So exposure information could be air quality, or if somebody lives close to a treatment plant of some kind, or they have a certain occupation that has an exposure. How we get those data and bring them together is going to be hugely challenging, but that's where OSU has such great expertise. Yeah.
And somewhat complementary to the next question, how do you see these models applied to common diseases or commoner diseases than rare diseases?
Yeah. That's a good question as well. And we've also recently started working on models for common diseases. The models for common diseases really require, so this is sort of very basic. It's a bag of phenotypes. You've got a set of phenotypes. We know when they start, we have onset and severity information and expressivity information, but really it's very brute force. It's just saying you have this collection of phenotypes and we can put them in a graph. For common diseases, we need a couple different things. We need to be able to have a sense of temporality and causation. So, for example, if you have diabetes and it leads to blindness, we need to understand the causation between those two things. So one disease can lead to another. Our definitions of what's a phenotype and what's a disease get much more blurred. And so the informatics tools aren't used to that. We have to these types of informatics tools are not used to that. So we have to think about how that works. We also have to think about much more complex genetics underlying those types of things, and much more probabilistic models, which we have some algorithms that use probability in these types of graph models. So we're pretty excited about it, especially we're trying to support some cancer diagnostics now that are much more aligned with that kind of an approach, but it's still pretty early days. But that's exactly where we're heading. Yeah. Yeah.
What do you see as the major challenge to deployment into clinical care? And I'm going to make a guess. Getting clinicians to understand how to interpret probabilistic data, providing them with an interface that they can interpret appropriately. That's my guess of what it might be. Mm-hmm. I'm interested.
Well, there's a number of different answers to that question, actually. And my first one might not actually be the one that you're expecting, but the first one is EPIC is my biggest problem. I've been working with it for a while now, and it's challenging because they like to do things their way and not anybody else's. But I think that there is the challenge of how do you train a clinician to think like a biologist, because that's what we're asking them to do here. We actually work a lot with the clinicians who are doing the phenotyping and have built different tools for them. The data visualization is really important. We've been building some new tools that kind of show probabilities and ask them what if they similar to when you're on Amazon, and it's very similar technology. It somehow knows when you're searching for one thing that you might be interested in this other thing that's not exactly the same. Those same kinds of technologies can be used to prompt a clinician to say, okay, if you entered these 5 things, here's the next lab that you need to do in order to see if you can get the differential diagnostic for that patient. So tools that help them reveal those things. And right now, most of those tools are very simplistic. They're list-based, not graph-based. So they're not looking across all the data. They're just looking at very simple definitions of disease. So using those kinds of visualizations are really important. The other thing that's just really funny, but just so true of all of us as human beings, is we started, and I didn't present this because it's a bit technical, but we have a tool that we call the annotation sufficiency metric, which is how good is your phenotyping. The people running the undiagnosed disease program asked us, how do we know when to tell clinicians that they are done, right? Like, when are you ever done? You could just keep going to describe patients, right? Many of them have. And so we implemented a system that takes any given set of phenotypes and looks against our whole corpus to say, how broad and deep and specific is a set that you have? We're not saying it's diagnostic, but just how good is it relative to the breadth and depth of what we already know is in the system? And we just put it on a 5-star system, and our annotations by the clinicians went through the roof, just with giving them gold stars. So it was miraculous, actually. So sometimes just having any kind of feedback is useful. And I think that, as we move forward, having more tools that help, kind of reveal what the differential diagnostics, how you can slim those things down in a kind of visually appealing way that's simple and quick as an overview would be really useful. The other problem is the genetic data currently in the labs that come back is just really hard for clinicians to interpret. So being able to look at genetic data and phenotype data together when you're looking to see what the differential diagnostic might be quickly without having to look at a whole genome and look at every transcript in that gene that you might be looking at all at once is just really overwhelming for many clinicians. So we need people who build video games to help us with this. That would be a good thing. So, yeah.
So I'm a layperson. Understood about the first 2 slides.
Okay.
Well, can I jump to the conclusion that through this tool, this process, that some currently diseases that are considered rare might be considered less rare as you diagnose and group people, and that could lead to more research funding, treatment options, and cures. Can I make that conclusion? Absolutely, and we're finding that already, that diseases that we thought were N of 1s were just actually much more prevalent. So it's I think in collaboration with the Stanford Healthcare Network, and in their network they're at a 10% diagnostic rate for what we know the rare disease incidence should be. So we're not diagnosing 90% of the patients that have a rare disease. So we don't really know, nationwide or globally for that matter, what the real incidence of these rare diseases really is. And those numbers are going to go all over the place. Some are going to go down percentage-wise, and some will go quite a lot up. But yes, absolutely. And I think, the other thing that's really interesting as well is we have different projects that I didn't talk about today that are really looking at finding modifier variants for a known Mendelian rare disease, in this case Fanconi anemia. And Fanconi anemia, as it turns out, has these modifier variants that really affect mutation and progression of the patient, but those modifier variants are very important for common diseases as well. And so we're revealing these underlying mechanistic
characteristics of these rare diseases is helping us understand much more common diseases, so we can think about treatments for not just the rare disease patients, but also for common disease patients. Is this the weirdest talk you've ever seen in the School of Public Health? Okay.
Very good, thank you.
Well, if you're interested, feel free to contact me. Thanks.